Hello. Hello. Yeah. So let me move on to the second session. Our speaker is online. It's my duty to introduce our second session speaker. Is Dr. Barath Mishra, having more than 14 years of teaching experience in pharmaceutical education with a proficiency in preclinical research, toxicological research, pharmacological screening models. Uh, He's always been active in preparing the students for future challenges by imparting an understanding of research and scientific writing. Sarah has already published almost 30 quality research articles and in conference proceedings in international and national journals, books. Is a trainer for soft skill programs and organized more than 25 conferences and seminars. He been a part of various reputed conferences as resource person in India and international level. Totally, he has backed more than 40 coordinatorship, and he also involved for the NBA process of our institution, Nirmala College of Pharmacy. Currently acting as a director of quality audit cell and head research and development. He has received grants from various government and non-government sectors, almost backed closer to 2 lakhs. With this brief introduction, I welcome Dr. Barath for this webinar to deliver his talk. His area of talk is COVID cure with reality check. So by this introduction, I'm hand over the session to Dr. Barath. Please stay tuned.
Presentation. So good morning, you all. I welcome again in this session the faculties and the student participants from the Nirmala College of Pharmacy as well as from the various institutions of Kerala and outside. I hope I am audible to all of you.
Yes, sir.
And yeah, thank you. So you all have received elaborative information about the background of COVID-19 infection and some basic information about it, as well as the information about the vaccines and the current treatment which is going on around. So my job is very simple, to take you to the in the treatment part, where I'm not going to tell you exactly what are the treatments and the things that are going to happen. You know very well, because these kind of things are quite easily available nowadays in everywhere. My interest is to discuss one or few very important contradictions or some controversies which have happened in between, and we must know about it, that how this all actually going on. So in that interest, I have brought this presentation for you. That's why I call it as a reality check. And by this, we will go exactly to find out that why these controversies have created, because you might have gone through last 3 months that various statement had been published by the Indian scientists from the ICMR, from the WHO, from the FDA, from the various other research organizations. So these all statements which have been published, either they published in the form of articles or in the newspaper articles, and there they have claimed various things, and they have been contradicted by various organizations. So where exactly we are standing now? My interest is to bring that information for you, that where exactly we are standing now, in spite of this all controversy which have happened. So these are some data already Dhanraj sir have discussed about it. Now you see there are in red color data which are taken by around 11th June, and this right side, the black colored data which are the 17th June, means yesterday's data. So these are the confirmed cases and the death by the WHO. So I won't go in detail of these things, just to brush you up. So I'll take you into the potential controversies. Here you can see the blue color spread all over the world. Here light blue to dark blue is the severity of cases. And here you can see the death rates from light brown to dark brown. It is gone through. So my interest is to take you to the potential treatments and then the controversies and the confusing statements which have been brought to the world, and how the the healthcare people, the healthcare professionals, the researchers, students, they all got stuck with these confusions. So I'll try to take little elaborate to this particular controversies which have happened. Let's see the possible treatments for now. Already Dhanraj sir has discussed about it, so I won't take it in detail about it. Just to mention in front of you is a kind of classification for now. This is the current classification what we are going through. So antiviral drugs which are currently in use are these drugs which are mentioned on the screen. As well as we have, these are pure antiviral class of drugs, and these antiviral class of drugs are also have lot many debatable discussions happened all around the world, that because they are the general antiviral drugs, and from the beginning they have been used in for various treatments. Even if you go for the remdesivir, you can find in the Ebola treatment they have brought first time to check for the Ebola, and they have been found some success, right? So the remdesivir has been used even for the Zika virus. They have tried and they found some success. Even for Nympha viruses, they have tried for the remdesivir. So like that, these drugs which have been used for various infections, viral infections, and so they are trying for the COVID-19 infection as well. And another combination what they have tried, the lopinavir and ritonavir. This is generally the combination because one improves the ritonavir improves the pharmacokinetic of lopinavir. So that's what they combine it and use it. So I'm not going in core pharmacology for all these drugs. And in this classification, you have anti-malarial drugs. Now, if you see the potential controversy what has been initiated around 3 months ago at the late February, around in initial time of March, and it has been ended. Even it's not ended because it's always a controversy is coming in, because the different researchers and their research organizations are bringing new information about it, and they some support it, some they go for opposing it. So anti-malarial drugs are there, and anti-malarial drugs where hydroxychloroquine and chloroquine always from beginning they have some role in the antiviral treatment. But in which kind of antivirus mechanism they are bringing in, that's always a debatable issue, and that's what they has brought a very potential controversy that I'm going to discuss. Other than that, you have some supportive agents. Azithromycin is there, vitamin C is there, corticosteroid. Somebody asked the question about dexamethasone because a recent article published on the dexamethasone. And what the article has published, actually, I have not found anything new about it, because we already know what the information about the dexamethasone has been used. So I will discuss on that issue as well. And nitric oxide is there, ipoprostenol is there, sirolimus, tocilizumab, and Canira. So these all are the drugs have been providing the supportive treatment for the antivirus because they are not the direct treatment for the COVID-19, but they are improving some of the symptoms, and that's what the ease of or the support the antiviral therapy. Some miscellaneous agents are also, and the therapies are also there. Angiotensin converting enzyme 2 receptor inhibitors and receptor blockers. This is one long talked issue is there. And in that also lot many contradictory statements have been published. Convalescent plasma therapy, which has been there, as well as some traditional herbal medicines have also been used. So now I'll move, and this is the slide, I think this already Dhanraj sir has shown you. These are the potential targets where these drugs are using. used. Now I'm talking about the confusion over hydroxychloroquine and chloroquine. I think some of the participants have already know about it, that what controversy has happened. But for those who don't know, I'm just trying to bring it, and this will give you, various facets about it. You can understand that how these things are actually happening. So what kind of confusion we are bringing in. Before that, we go into the confusion. I will try to tell you what the ICMR has given what called the guidelines from the beginning itself for the uses of HCQ and CQ. So they are telling that 400 mg HCQ first day we can give, and then it followed by the 200 mg HCQ for next 4 days, and then the total doses should not exceed more than 2,400 mg in 5 days. So this is what the regimen schedule what ICMR has proposed. Now, if you go into see the series of statements what have been published by the ICMR, WHO, FDA, and various other organizations, you will see the ICMR has almost most consistent stand on the HCQ, while the other agencies have quite variable or the fluctuating stand on it. Now we'll see one report has been published in the print in 25 March. I'm taking you to the few potential statements which have been published from beginning and what they have claimed. So around the March mid, the ICMR has given one advisory, and according to that advisory, they are telling that cautious use of hydroxychloroquine can be given. And initially they said that it will be good for the health workers. Later they have said it can be given for a little more asymptomatic people as well. So like that, they have issued consecutive statements on this particular thing. And in the ICMR has also said this uses of HCQ for the COVID-19 as experimental. So they have bracketed this word experimental, and they said this is an experimental now. It means they were not sure that how it will move, but they have tried it for this. In the March end, FDA also has given one statement. They said that emergency use authorization. So means COVID-19 can be used. They have just little extended it that for the hospitalized patients and serious COVID-19 infected patients, the emergency use of the HCQ can be done. So like that, statement to statement, it is moving. Now I'll take you to one potential conflict between the ICMR, the Lancet, and the WHO. This is very important to know for all the researchers. That's why I thought to bring it here. So what happened? I'll take you the series of the discussions what happened here. First, I will tell you the paper published by the Lancet in the May 22, and what they have quoted. They have quoted that the hydroxychloroquine, often in combination with second-generation macrolide, are being widely used for treatment of COVID-19, despite no conclusive evidence of their benefit. So what they are claiming here, that there is no conclusive evidence is there for the use of hydroxychloroquine. And they said that yes, it is a very good drug or proven drug for the some autoimmune diseases or in the malaria, but in the COVID-19 is very much confusing. So like that one statement published in the Lancet through a one paper. So I will give you a little more information on it. It is published by May 22. Around May 26, WHO has boldly given one very crucial statement. What the WHO has said: although hydroxychloroquine and chloroquine are already licensed products for treating other diseases, at this stage, these drugs have not been found to be effective in the treatment of COVID-19. So it was a big statement, because ICMR has confidently were using it, and they are almost all over the world. The hydroxychloroquine treatment was started, and a big amount of drugs has been exported from India to all over the world. So this statement was quite disheartening for us. And then what happened next, I will tell you, because it's a one big story. So, and as well as the WHO has said, given a warning to all the hospitals that there should be very limited use of the hydroxychloroquine. And later what happened when this controversy rises, the WHO has started one solidarity trial. Solidarity trial is one individual trial what the WHO has started, but it was a very, very big, because 400 hospitals in 35 countries are actively recruiting patients. At nearly 3,500 patients have been enrolled for from 17 countries. So it's a huge trial what has been initiated by the WHO. And in this trial, one arm of the trial was dealing with the HCQ. So I want to tell you this is a very, very important thing, because WHO was doing on the world level this trial, and one particular arm was dedicated for the trials on the HCQ. What happened when the Lancet published that paper? WHO has quoted the Lancet paper in their advisory, and from here the controversy begins. So what the WHO has said, as an example, an observational study published in the Lancet on 22 May found that among 1 lakh patients from multiple countries randomized to receive hydroxychloroquine, when used alone or with macrolide, there was a higher mortality rate and an increased frequency of irregular heartbeats. So this was the biggest statement what the WHO has announced. And after this, the huge controversy started all over the world, because WHO has quoted one paper from one publishing house, and resultantly, what WHO has done, WHO has temporarily paused the hydroxychloroquine trial from the solidarity trial. They have stopped one arm where they were doing the solidarity trial, and that was the biggest problem, because they have stopped doing all that thing, and they said what the Lancet has published, we are believing on it, and we are stopping all our what called the clinical trials on it. Then what happened? What they have declared? They said we are, COVID-19 patients enrollment for this drug was temporarily suspended on 24 May. That was the biggest statement by WHO, and they stopped it literally the clinical trial what they were begin with. Now, once the Lancet published, the controversy came, and then WHO has taken it seriously and stopped the clinical trial. Then controversy has even get deeper and deeper. And now what happened next? WHO has said, released one advisory. In fact, up to 2 days before that advisory was there in the WHO website. Maybe today also, I have not checked today. You can find it out. So what they stated, unproven treatment. They said that everyone has to stop this unproven treatment, and to discourage the people to take it as a self-medication also. So it means India, which is the biggest producer, India, which is the biggest exporter of the HCQ, got stuck here. It's a big thing. What WHO has finally said in this report, he said he stopped the use of HCQ, and what he said, that based on data from laboratory and clinical studies, remdesivir, lopinavir, ritonavir, and lopinavir, ritonavir with interferon beta 1 alpha can be given, while the HCQ can be selectively used here and there. So like that, what the WHO has said. Now, in this particular statement, I tell you, I want to quote one more controversy which has initiated during the March. One paper published in the New England Journal of Medicine, and they said that the uses of the anti-HIV drugs for virus COVID-19 is not at all a right choice, right? And that paper has also been discussed a lot during that time. Later, some other studies published, and they supported what WHO has said. Now, I'll tell you what ICMR took the stand on it, right? Now, this is a big controversy happened. So what ICMR has said, the Indian Council of Medical Research continued to recommend HCQ as a prophylactic measure against COVID-19. I tell you, when the advisory was published in the WHO website, even that time, the ICMR has given the advisory to continue the use of HCQ. It was a conflict between the ICMR and the WHO. And then a lot of paperwork was going on. So they have declared it and continued the use of this one. One very, very potential report I wanted to mention here. You can go in detail and read about it. The link is given here. Published in the Dental Tribune, what they said, it is published in June 1st, this month itself, June 1st, what they published. new case study, new control study by ICMR shows hydroxychloroquine can prevent COVID-19 in healthcare workers. Now you can see, one side WHO has given the advisory and they are telling stop using hydroxychloroquine, and one side the ICMR has published the advisory and said continue using of the HCQ in the health workers with the with supportive measures. So this they have given. What I want to quote what the ICMR or the papers, other researchers have said to Lancet paper. What is their take on the paper published in the Lancet? They said more than Lancet faces severe criticism from scientific community. So one thing for that paper, which is published and quoted by WHO, was facing severe criticism from scientific community. And they said the data has been, they are hiding the data on hydroxychloroquine, right? So more than 100 scientists and clinicians have sent an open letter to the Lancet's editor, Richard Horton, and the paper, authors asking them many uncomfortable questions, but have failed to receive convincing answers from the authors. So what happened? When all over the world, the questions are flooded to the Lancet, and the Lancet was sending the questions to the authors, and authors may not have handled the questions well. So, and if you want to read that open letter, what the people, the 100 the scientists have sent, in this most of them are from India as well. And this letter link is given here. You can click on the link and you can find that open letter. Open letter is given there, and you can find what are the uncomfortable questions asked to the authors of the paper published in the Lancet. Now, after that, what ICMR said for the trial, the control study, what ICMR does, ICMR has declared it and said that in their study, only 3 kind of side effects have been observed: nausea, headache, and diarrhea. That is also 8%, 5%, and 4%. And in some cases, they found some palpitation were there. While the Lancet paper has said the serious cardiovascular symptoms, cardiovascular ADRs have been observed, right? As well as some gastrointestinal 0.6% has also been seen. And some cases 0.6% and control 1.4% had skin rashes. So this is what concluded by the ICMR, right? And ICMR found there is all these adverse reactions what has been presented here, nothing is so serious and life-threatening. Now, what the Indian officials have said on the Lancet, that paper, what they said? They said, Indian official disagreeing with the WHO's advisory believe that the doses level given in the international trials are massive, 800 mg into 2 loading doses for 6 hours, a part of 400 mg into 2 doses per day for 10 days. So the total dose what they were giving, that is 9,600 mg given over 11 days. So now you can see that how much big ratio was there, and they found this is a mistake in that. And because of that, they are not saying that that is a fake report, but they said that the trials have been done wrongly. Maybe they have not used the proper things, and that's what they have pointed out in the doses. So if you like to read, this is a very interesting controversy. That's what I thought to bring it for you. And if you want to read the entire statement of series of statements what have been initiated from beginning and to that, there is one report published in the Hindu, and that link I have given here. Just click it and find all these statements given there. Now, okay, so this is all about the controversy which has happened. Now what happened? What is the current situation? So current situation is somewhat relaxing or comfortable for us, that finally controversy ends for now. Right? I'm writing for now because I don't know what other controversy is going to come. But right now we have the controversy ends. How it ends? Now I'll tell you how it ends. The Lancet, after this much criticism all over the world, Lancet has retracted that paper from their list, right? And it means the paper is retracted, paper is still available, but it is having, you can see in the screen, it is having a big font, retracted. So the paper has been retracted from it. Now, what happened? because the Lancet paper was the basis for WHO to give that advisory. Now, when the paper has been retracted, what the WHO has to say? Now, you see what the WHO has said. This is the statement of WHO after the paper has retracted. This is, I'm talking about just few days ago. WHO restarts HCQ trial after Lancet concern over paper that trashed it. In the statement, the Lancet said, now I'll tell you what the Lancet said on that statement. Important scientific questions were raised about data in the paper authored by cardiologist Mandeep Mehra, Desai, and Zurich cardiologist Frank. It is said an independent audit has been commissioned. So these authors, now the one big problem is going on for them, right? And these authors are not from the very small institutions. One is from the Harvard, and one is from the Salt Lake University. One is from the, another is also from the USA. Desai is from the USA. And one is from the Switzerland. So all are from very, very big universities, what they have done. And now they are facing a problem because Lancet said independent audit has been commissioned for this particular thing. So now the WHO has nothing else to say. So WHO has taken that advisory back. If you want to read this entire report, the link is given. You can click it and find it out. This is the link. These are the quick tweets what the Dr. Tedros have sent after the Lancet has withdrawn. What they are saying, the executive group received this recommendation and endorsed continuation of all arms of the solitary trial, including HCQ. So now they have again resumed the clinical trial for the HCQ, and they have taken back that advisory. So what is the conclusion out of it? We have seen that how the controversy has gone. Now how we can conclude over it. So I will conclude what the ICMR said, because ICMR is the one which is more consistent for his statements from the beginning. So what the ICMR has said, this is we can take as a concluding remark for this. The public health message of sustained intake of HCQ prophylaxis as well as appropriate PPE use need to be considered. Now what they have added one more clause in this, that whenever the people, those who are using the HCQ must use the PPE kit along with it. So along with these two, they are telling is going to be useful. HCQ as a primary choice for prophylaxis against SARS-CoV-2. That is, they are still maintaining their statement. And what they said, that they have given two major reasons why they are, bystanding with their statements. First, the ability of HCQ to inhibit the infection of SARS-CoV-2 as well as viral replication in cell cultures in a time and dose dependent manner. This all have the references for this. Second, the effects of HCQ to elevate the pH of endosomes and inhibit SARS-CoV-2 RNA-mediated inflammatory responses. So these are the two basic reasons by which the ICMR is supporting strongly to the use of HCQ. So we can take this as a back home message that all the controversy which has happened, but we are sticking on it because of these two strong reasons. Okay, now, so this is here, you have seen the controversies, and now you have seen how it has been ended. And these are very important papers. I think you all have to read it. As well as for this Lancet paper, even it is retracted, but you all must to read it to understand one more very important thing, that all the researchers should take that responsibility whenever they are publishing the paper. They are giving a very potential information, right? And if you are hiding some data, and if you are doing some kind of thing, maybe you can face an international level inquiry against you. So this is a one learning for all the researchers from this paper as well. Now, I will take you that to another confusion generally people have, that it is used for malaria, then why it is used for COVID-19. So what exactly it does for the COVID-19 virus, while it is an anti-malarial drug. So I'll take you just a few information about it, so you'll make it understand why it's happening here. When we go for the mechanism, we can find these four potential mechanisms what HCQ does. One is immunomodulatory effects. Usually, this immunomodulatory effects is associated with the use of HCQ for malaria. But alkalinizes lysosomal pH, the second mechanism. Third is HQ is a zinc ionophore, and it binds to sialic acid. So these are four. So second, third, and fourth mechanisms are very, very potential for the antiviral use of the HCQ. So here I will tell you this mechanism by which you can understand well. First thing, the first mechanism, which is immunomodulatory effects. So there, because I am discussing about COVID-19 mechanism, so that only I have taken in detail, but I'll just mention for it that HQ inhibits the degradation of cargo. Cargo means when the proteins, the virus enters into the lysosome, it get a combination with the protein and the organelle that called cargo. So in autolysosomes, by increasing the pH to prevent the activity of lysosome enzyme, inhibition of lysosomal activity can prevent MHC class 2 mediated autoantigen presentation. So that is what the immunomodulatory effect it is producing and being used for the anti-malarial treatment. Now the second, which is we are discussing here, alkalinizes lysosomal pH. This is the one mechanism what the antiviral the scientists have been encouraged to use it for the antiviral therapy. So you have seen already the here the virus when enters reaches to the cell membrane, it uses the ACE2 receptor to bind with. And once it binds it with, it go for the endocytosis, right? Most of the time you see the endocytosis process happens whenever microbes are entering. So in the endocytosis process, the whole virus particle is getting entrapped into one endosome. Now later, this is the first step. The second step, what is happening? This endosome is going to combine with the lysosome which is present inside the cell, because lysosome has lot many lysosomal enzymes. And these lysosomal enzymes are very important to uncoat the virus particle and to release out its DNA. So in this case, this is very important fusion where the lysosome is going to fuse with the endosome. And when they fused it, they form it endolysosome, right? So now what is happening here? The chloroquine, when enters into it, it is first thing, it inhibits this endosome and lysosome fusion. Right. Another thing, what the potential it is doing, it is changing the lysosomal pH. It is rising the lysosomal pH. And while because it is rising the lysosomal pH, the lysosomal functions get infected or the lysosomal functions get affected. So lysosome won't do what it has to do, right? So endosomal and lysosomal functions get subsided, right? And then the further action of the what's called the viral uncoating and the multiplication and that all process already get hampered by it. So that what the chloroquine is doing here. One more thing what chloroquine does here, you can see in the molecular mechanism, that once it increases the pH in the lysosome, there is a toll-like receptors, right? These toll-like receptors get activated when in the normal process, and the signaling process have started. So usually TLR7, TLR9, TLR17 as well as TLR22, they all are the the receptors are there which get activated when the virus enters into the lysosome, and then they generate a process of communication. That process has stopped when the chloroquine changes the pH inside the lysosome. So like what it does. Another mechanism what the chloroquine also does here, by preventing TLR signaling, it also interferes the GAS stimulator of interferon genes. So there also the signaling get affected, and hydroxychloroquine can reduce the production of pro-inflammatory cytokines. That actually what it does. So not only the it infects what called affected the functioning of the lysosome, but also it inhibits the pro-inflammatory cytokines. So it reduces the inflammation process as well. You can see here, in this case, HCQ is entering into the endosomes, and there it is going to bind with the TLR, TLR7, TLR9, and then it activates the TLR signaling, right? This TLR signaling goes to, produce the expression, or you can say activates the expression of pro-inflammatory mediators, right? The second mechanism, you can see that it acts on the CGS, and also it inhibits the CGS string signaling. So ultimately, it is what it is doing. It is subsiding the communication pathway by which the inflammatory mediator is generated. Another process what the chloroquine does, that it facilitates the movement of zinc. That's why they are called as zinc ionophores. So they are facilitating the movement of zinc from outside to inside. Now what this zinc does when it enters into the cell, what it does, this particular zinc, because in the normal process, what happens when the lysosome, when the virus enters into the lysosome, it started replicating, right? And it forms its own mRNA. And that mRNA, by using the RNA-dependent RNA polymerase, get multiplied. And then these viral RNAs, many viral RNAs which are formed, they get into the what called the packing, and then they form the new viral particles. So what the zinc does here, zinc interferes in the RNA-dependent RNA polymerase to make more copies of viral RNA. So it's a double effect of the chloroquine comes here, right? Directly chloroquine also, by increasing the lysosomal pH, it works. Second, it works by facilitating the zinc movement from outside to inside. This is the two mechanisms where the chloroquine acts. So I hope this is clear what it does into the COVID-19 treatment. Right now, we'll see what it does in the malaria. Right. So here it is used, for what it does. It interferes with the lysosomal activity, apoptosis, autophagy, interacting with the membrane stability, and altering signaling pathways and transcriptional activity, right? So, and potentially what it does, interferes with the formation of hemojoin pigment. You all know, whenever the hemojoin pigments released out, they are actually causing symptoms of the malaria, where the what called the shivering and fever started coming in. So it is this drug preventing the hemojoin crystallization, right? So you can see in this diagram here, where you can see the right side one diagram is there, where you can find that when chloroquine enters, it gets associated with the H+ ion, and this protonated chloroquine is able to inhibit the heme's polymerized formation. So polymer formation will not be there. So hemojoin granules which are formed few won't affect that much, and that's what the chloroquine does. So these two diagrams are enough to explain you that how it is doing. Okay, so this is about the HQ. I think from beginning the controversy to the confusion on the mechanism, I tried to make it here. Now I'll take you to the another very, very important drug, that is remdesivir. So what remdesivir does and what the remdesivir is actually used for. So remdesivir is a phosphoramide prodrug of an adenosine C nucleotide, right? And it has been used, as I told you, for the Ebola treatment also. And remdesivir is metabolized into its active form. So remdesivir is acting as a prodrug. When it goes into the body, it converted into its active metabolite, right? And that is actually doing the job. It obscures viral RNA polymerase and evades proofreading by viral exonucleases. So I'll tell you what it does and how it does, causing a decrease in viral RNA population. So how it is happening? By this mechanism, you can understand well that the virus when enters to ACE2 receptor goes in from the endocytosis and makes the process already, through endocytosis make the endolysosome, and through that endolysosome the viral get uncoated and the viral RNA comes out, and this viral RNA utilizes the RNA dependent RNA polymerase and to make more viral RNAs, and then these viral RNAs get packed into it and they do the job. So what your drug is doing here, you can see the remdesivir. What is doing here? This remdesivir is entering and converting into its one potential metabolite. You, especially students, you keep remember this number, because this is the metabolite product of the remdesivir, GS441524, right? So this remdesivir converted into its active metabolite, and this is this one. And this metabolite goes and interfere in the two processes. One, the formation of the more RNA particles. Second, while the viral RNA particle form, it is also forming 3-5-exoribonuclease. This is what? This is a proofreader for the RNA which is going to form. So when the remdesivir is given, it is interfering in the proofreading process as well. So it is interfering in two processes. One, the formation of the more means activity of the RNA dependent RNA polymerase to make more RNA particles, as well as the proofreading process of the new generated particles. So like that, remdesivir act in this particular process. Now, somebody has the question on the dexamethasone. And dexamethasone, yeah, sorry. So, recently, one article published, and I think that's why the dexamethasone discussion came in picture earlier. Otherwise, the uses of corticosteroids was there along with the treatment from beginning, because dexamethasone or the other corticosteroids are the providing an associative treatment for it. What dexamethasone does? Dexamethasone reduces death in hospitalized patient with a severe respiratory complication on COVID-19. This is a statement, right? Who has given? One professor named Peter Harvey from the University of Oxford. They have given this on 16th June, means 2 days before this paper published, and they have given this statement. Dexamethasone is the first drug to be shown to improve survival of COVID-19. This is an extremely welcome result. I didn't understand exactly why dexamethasone they are highlighting it too much, because dexamethasone or other corticosteroids are very much used from beginning, and their job is just to reduce the excessive release of the interleukins, right, and other cytokines. So because these cytokines are producing many serious symptoms, so this is dexamethasone is not at all a cure for the COVID-19. Just underline this statement. This is not a cure for the COVID-19 treatment, but it is a supportive or adjunct therapy to subside certain serious symptoms. See, these, because the respiratory tract get infected by the virus, and because of that, a lot of inflammatory mediators released, and they worsen the patient's situation. So what dexamethasone does, these steroids have been very well established for their anti-inflammatory effect. Here also we are getting the same, right? So it's nothing, not a big deal to say that this is big statement, but they have done the trial, and they have for the COVID-19. That's why this paper has been published. You can find the details in the below link given. Now I'll just take you 2 more minutes to explain you about the tocilizumab. What it does, it's also having the similar kind of effect what the steroids are doing or other drugs are doing. Here also, whenever the interleukin get bind with it, they're producing many inflammatory situation. And tocilizumab is also blocking the binding of the interleukins with their receptors, and they are calming down or reducing the inflammation. That's why they are all subsidiary for the viral treatment. Azithromycin, it is also act for the respiratory syncytial virus. It has been, azithromycin has been also used from long time. During the Ebola and Zika virus, they have used a lot during that time, because it is also having a slight, along with the antibacterial activity, it has some antiviral activity as well, because it is acting on the pathway of the Rho kinase pathway. One more serious discussion was there on the angiotensin converting enzyme 2 receptor inhibitor. Just I am taking few lines to make you clear what actually it's doing. So first I will tell you what the ACE2 was used for. So ACE2, what it does for our body, it is regulating the blood pressure, wound healing, and inflammation processes. Okay. Now, ACE2 counteracts the effect of ANG2. Now, this is a very important point. Angiotensin 2 has some toxic effect in our body, and these effects can be controlled by ACE2. And this is the same ACE2 which has been used by our virus to bind with it. If you want to subside the ACE2, then ACE2 effect will be subsided. Then what about the toxic effect of ANG2? That's why ACE2 cannot be subsided. Otherwise, ANG cannot be controlled. Right? This is a problem by subsiding the ACE2. So harmful effects of ANG2 are reduced by the ACE2. That's what actually ACE2 does. SARS-CoV-2 virus bind to ACE2. It prevents ACE2 from performing its normal function to regulate the ANG2 signaling. Okay. So if SARS-CoV-2 binds on ACE2, then the ANG2 won't perform its normal functioning, right? ACE1 increases the amount of angiotensin 2, whereas ACE2 reduces the angiotensin 2. So our purpose is that ACE2 is needed for the ANG2 regulation, but if ACE2 is there, virus will bind and go into it. So this is a one dilemma is coming into it in one area. Here, all the references have been quoted. Everything, all the statements are from the all well-reputed papers, and mostly from the WHO and ICMR websites. You can click any of the link, and these are very, very important references. You can go through with it. Maybe a treat for the researchers to go for it, right? So I hope I have cleared you all the controversies as well as the confusion among the drugs. And thank you for your patient listening. And I hope I have done some help to you to understand these things for you. Thank you.
Thank you, Dr. Barath. Hello, hearing? Thank you,
Yeah, I
Dr.
can
Barath.
hear you. I can hear you. Yes.
Yeah, I think Speaker Dr. Barath Mishra justified his topic, and he's taken up his talk from the huge controversy cases, right? And against the COVID drug, and very, very informative and narrative too. There is no doubt, and he has given clarifications regarding the ICMR statement and WHO statement against hydroxychloroquine. And also, one of the best paper being retracted from the Lancet. This is also a point to be noted. Thank you very much, Bharat. And he's also highlighted about hydroxychloroquine, its property, antiviral therapy, and its mechanism. They also touched about remdesivir. You know, it's all been used for the treatment of Ebola virus for the infected body. And he explained about its metabolites, how it acts and possesses an active antiviral property. And touched about dexamethasone, azithromycin. As last, he has concluded with the ACE2 and ANGH correlation, very well explained. So over to the participants who are interested or who are in the position to raise a question now, you can. Over to the participants. I request any of the audience wanted to put their questions towards our speaker is online. Okay, there's no questions in the chat box also. So as a moderator, I just wanted to know you one thing, sir. You used the word that azithromycin acts as a antibacterial as well as antiviral. So can you highlight a mechanism or a line of message how this drug acts dually for antibacterial and antiviral?
So reason is that whatever the microbes are there, right? Either it's a bacteria or viruses, their binding mechanism through the cells are almost similar, and their inside, when they enter into the inside, the treatment of cell towards them is also the similar. So what azithromycin does, because its mechanism, these macrolides are, that's what the WHO also said, that macrolides are used with the SCQ also. So what they are doing, they are potentially, delaying the process of the chain termination, means the chain sealing. So when the what's called nucleic acid chain, when gets elongating for the RNA, and it's not getting sealed on properly, the virus get inactivated. So this is the way in the bacteria as well as in the virus, it does the similar way effect. And that's why, and some effect, because along with the SCQ, when we are giving, it is also potentiating what binding of the virus, as what I have already explained. So this way, azithromycin is somewhat useful for treating it. It's not it's a first line treatment. It's just an adjunctive treatment what we are giving along with SCQ to potentiate that. That's all.
Okay, thank you, Dr. Bharat. And I think we end up with the second session. And thank you very much, participants, for patient listening. And some of them have really interacted with our speaker. We are just winding up this today's webinar meet. Thank you for joining with us. And behalf of Nirmala College of Pharmacy, we request you to give your feedback. The feedback link will be shared on the chat box.
So I would like to thank you to moderator, to the Nirmala College of Pharmacy, management, principal sir, for creating this webinar series, and I'm very much thankful being the part of it. And all the details what I have given here all are referenced. So anyone have any doubts, please go to the references and please find it. Thank you so much.